Hello students, a warm welcome to all of you in IT Life Academy. In today's lecture, we will discuss about the introduction of clinical microbiology. Basically, in today's lecture, we will discuss about the overall idea of clinical microbiology. In today's lecture, we will discuss about the in today's lecture, we will discuss what is the clinical microbiology and what is the role of clinical microbiology to diagnose a particular disease. Okay, so we start today's lecture and understand the today's topic. First, we understand what is the clinical microbiology. Clinical microbiology is a branch of medical science. Branch of medical science in which we study about the role of microorganisms. Role of microorganism like bacteria, like virus, like fungus, parasites and mycobacteria. In clinical microbiology, we understand the role of these microorganisms to cause a particular disease, to cause a particular to cause a disease in human beings. In clinical microbiology, we study how these microorganisms cause a disease in a human, how we diagnose this particular disease, what is the mode of transmission, what is the treatment of this particular disease, and what are the method by which we control this disease to spread between humans. Okay, and the role of microbiologists to understand how the microorganisms cause the disease means we understand the mechanism, the mode of working to cause a particular disease, and how this particular disease is transmitted, and how we control this particular diseases. This is the role of clinical microbiologist. Or what is the clinical microbiology? Clinical microbiology is a branch of medical science where we study about the role of microorganisms to cause a particular disease or to cause a infection. Understand the Now we understand the function of clinical microbiology laboratory. First is a sample collection. Okay, and this sample may be blood sample, maybe urine. Okay, then next step is a labeling. Why? Patient name, or by any number or keyword. Okay, after the labeling, next step is storage. Storage according to the sample. Some sample are stored at a cold environment, some at a room temperature, and some at a high temperature. Okay, then next step is prepare for the analysis. Prepare for the analysis. Okay, then examine. For examine, we use microscope, maybe light microscope, maybe electron microscope. Okay, maybe staining method. Staining like gram staining, like acid fast staining. Okay, gram staining for bacteria to understand the gram positive bacteria or gram negative bacteria. Okay, these are the methods. After this method, isolation of microorganisms. For isolation, first we perform serial dilution. After the serial dilution, we perform the process of spreading on a culture media. Culture media, for example, nutrient agar. After the spreading, sticking. After the sticking, we isolate the pure colonies. Okay. Okay. Sometimes, if our microorganisms are unknown, then we follow the Verghese manual, and in Verghese manual, we perform a lots of biochemical tests. Lots of biochemical tests like catalase test, like coagulase test, oxidase test, mannitol test, BP test, etc. Other than this, we apply some automatic method like BitTech, like MALDI-TOF and mass spectroscopy. Other than this, we apply the some microbiological method like PCR, that is a polymeric chain reactions, or nucleic acid probe. Function is a antimicrobial sustainability test. Next is a antimicrobial Next is a antimicrobial susceptibility test, in which we determine the sensitivity of pathogen to various antibodies. Okay. For this, we apply a two method. First is a disk method and MIC determine method. Okay. And guide the clinical in an effective therapy decision. Okay. This technique is very helpful. Okay. Then serological diseases testing. In serological testing, and we detect the antigen antibody reactions, antigen antibody related infection. For this, for example, like HIV, HBV or dengue. For this, we apply the technique like ELISA, latex, latex agglutination, latex agglutination. Okay, molecular diagnostic technique like PCR to detect the pathogen for the rapid diagnosis, to detect to detect the resistance gene or epidermological typing. Next is the infection control and hospital surveillance. Okay, we identify the hospital acquired infections. Okay, and we also monitor the outbreak and supporting infection control. Epidemiological surveillance means tracking the prevalence and threat to infection disease in the community. Reporting the notifiable. Next is the epi. Next is the epidemiological surveillance means tracking the prevalence and trend to infection disease in a community. Reporting the notifiable diseases to the public health authorities. Next, quality control and error and assurance means we ensure the accuracy and reliability of the testing result. Next, regular collaboration, validation and participation of external quality assessment schemes. Next, research and development, develop the new diagnostic methods. Okay, and study the microbial resistance pattern and emerging pathogens. Education, training the microbiologist, lab technician, intern, etc. And updating the staff with new diagnostic therapeutic techniques and infection control practices. Okay, this is the role of clinical microbiologist lab. Now we understand the common techniques used in clinical microbiology. First is a microscope. Microscope is used to visualize to visualize the microorganisms. Okay, and microscope are divided into two parts. It may be electron microscope, it may be light microscope. Electron microscope like SAM, like TAM. Okay, light microscope like dark field microscope, fluorescence microscope, confocal microscope, bright field microscope, etc. Okay, light microscope, gram staining, acid fast staining and wet mount. Dark field for the for spirochetes. Fluorescence microscope using the fluorescent dye to visualize the microorganisms, and this is very useful for the diagnosing the Mycobacterium tuberculosis bacteria. This is very helpful for diagnosing the Mycobacteria tuberculosis. Electron microscope to study the structure of viruses and small bacteria. Okay, these are the microscopic techniques. Next is the staining techniques, gram staining and gram staining. This technique will differentiate the gram positive or gram negative bacteria. Acid fast staining, this is for the Mycobacterium species. Capsule staining to detect the capsules. Endospore staining to detect the bacterial spores. Simple staining, methyl blue or crystal violet for the morphology. Okay. Now, next is the culturing technique. The purpose of this technique to grow or isolate the bacteria. Media. First is a media in which our microorganisms will grow. This media may be solid media, may be liquid media, may be selective media, differential media, defined media, undefined media, etc. Okay, solid media like nutrients agar, blood agar, and Marconi and Marconi agar. MEC कौन की आकार. Okay, liquid media like enrichment of low number. Selective media contains the inhibitor for the unwanted microscope, that is a mannitol salt agar media. Differential media, differential bacteria based on a biochemical property like MECCON के differentiating lactose fermentation process fermenter. Like differential media differentiate the bacteria based on a biochemical property like MECCON की differentiating lactose fermentation for lactose fermenter. Okay, these are the different types of media which we use to grow the microorganisms. In lab, most of the time we use nutrient agar media. Next, biochemical test. It will like catalase test, coagulase test, oxidase test, indole test, methyl test, BP test, citric acid test, urease, triple solid iron test. These are the different types of test. Okay. Next, serological techniques like ELISA, that is the enzyme linked immunosorbent assay, lattice agglutination testing, ELISA, lattice lattice agglutination testing, immunofluorescence assay, and Western protein. Antibiotic sustainable tests like DISC method, E-test, broad dilution method, and automatic system like BitTech and BD-Phenox. Okay, these are the antibiotic sustainability test. Okay, and this portion will repeat it. Next is a molecular biology test like PCR, that is a polymerase chain reaction, RT-PCR for the RNA viruses, DNA sequences to identify the exact sequence of the DNA, hybridization technique like FIS to identify the desired gene. Okay, automatic identification technique, and the purpose of this technique is it is very rapidly identification and sustainability testing. Example, Malditov, MS, Vitec2 and PD-Phenox. Immunological detection purpose detecting the specific antigen or antibody. Example, rapid test of for malaria, dengue or HIV. Virus culture to isolate the virus culture using a different cell line. Use of Vero, HeLa, Medex cells for influenza and other viruses. Mycological testing, mycology testing like direct microscopy, parasitology technique like wet mount, iron mount, concentration technique. Aerobic culture technique, bacteria grow in anaerobic condition. Quality control and quality assurance, ensuring the accuracy, reliability of all the tests, regularly cultivate and control strain. Blood culturing techniques like BAC-T and adult for the rapid detection. Okay. Now these are the techniques and their role. Okay. Now we understand the scope of clinical microbiology. First is to identify the pathogens, identify, isolate and identify of bacteria, viruses, fungus, parasite. Diagnosis of infection diseases, laboratory testing to confirm the causative agent. Antibacterial, antimicrobial sustainability tests to determine the sensitivity and reactance of a microbial antibiotic. Guidance and physician appropriate therapy. Infection control means surveillance to hospital acquired infection and implement of infection prevent strategies. Public health outbreak investment, reporting the notifiable 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 infection diseases. Okay, now these are the different division of clinical microbiology, like bacteriology to study the bacteria, virology to study about the viruses, mycology to study about the fungus, parasitology to study about the parasite, immunology to study about the immune system of humans, molecular diagnostic use the RT-PCR and sequencing method for the pathogen detection. Now we understand the step-by-step process of clinical microbiology lab. First, specimen collection. This specimen may be urine, maybe blood, maybe others. Proper labeling and transport. Then microscopic examination, gram staining, acid fast staining or KOH staining. Culturing, for culturing, or we use a specific media for the appropriate condition. Okay, for the colony morphology, pigmentation and hemolysis pattern. Then biochemical testing like catalase, oxidase, coagulase, fermentation tests. And finally, we identify our bacteria. For biochemical testing, we follow the rules of we follow the step of Burgess-Malloy. Next, serological testing detect the antigen and antibiotic tests like ELISA, and molecular biology technique RT-PCR, PCR, nucleic acid, etc. Antimicrobial sustainability test like DISC method and like MIC determination methods. Okay, this is the scope of clinical microbiology. Role, interpretation of lab result, advising the clinical treatment and infection control, implementing the laboratory specific standard, contribute to hospital infection and control community, and training lab staff and students. These are the role of clinical microbiologist. Importance of clinical microbiology: accurate diagnosis, appropriate treatment and choose, control the infection and disease spread, surveillance of antimicrobial testing, public health management. These are the importance of clinical microbiology. Recent advancement in clinical microbiology like MALDI-TOF, this is a mass spectroscopic technique. This technique is very useful to identify the bacterial. Next generation sequencing in pattern genomics. Automatic blood labeling like point of care testing and bioinformatic applications. These are the RADBIT. This is the recent advancement in clinical microbiology. Next is the clinical path, career path in microbiology. First, clinical microbiologist or medical microbiologist, medical laboratory scientist or medical technologist specialized in microbiology, or research scientist and research scientist. This is all about the today's lecture. Please follow ITLS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and also get it on Google Play Store. Thank you so much for watching my video. Please like, share and subscribe ITLS Academy. Thank you.